Okay, so welcome back, everybody. This is the second webinar of today. So this webinar is going to be about some of the product development pathways and strategies that some ATMPs have taken for commercialization. So this should be complementary to some of the other webinars that we will be doing. So it's not going to be an overview of ATMPs at all. It's going to go straight into some of the products and what they are. So the aim then of this is just to really summarize using two models, some of the regulatory and scientific pathways and decisions that were made to get onto the market in the EU. So we're not going to revise in any detail any of the regulatory procedures, how they work, what they are. We assume some knowledge of that. And I think Joel is going to talk about that in a couple of weeks' time. So if you're not so familiar, we can look out for his webinar when it comes up soon. So the focus will also be on the European approval pathway, not so much FDA, just for time's sake. So I'm going to give a one-slide overview of drug development in general, just to put some context about some of the things that we'll mention for the products. And the two examples that we're going to talk about today are Strimvelis, which is for the treatment of ADA-SCID. So this can also be considered an analogous model to CAR-T, for example, because it's an ex vivo gene therapy. And also Luxturna, we're going to have a look at that, which is for treatment of inherited retinal dystrophies, okay? So here's our one slide overview of drug development, okay? So at the top, got different timelines. These are estimates based on small molecules, and then different phases of development, a kind of discovery phase, development phase, which is a more formal pharmaceutical development phase, and then obviously post-authorization commercial phase. Okay, so maybe at the beginning you're going to start out in the laboratory, in a research group, in a university, for example, and then you will basically develop a concept for a gene therapy vector or a cell therapy or whatever, and then you'll decide to take it into preclinical research, safety, toxicity studies, and so on before doing clinical research, and then prepare an MAA, and then obviously you'll have some post-marketing commitments probably as well. So the clinical phase usually will have phase 1, phase 2, and phase 3, and you might have phase 4 post-marketing studies as well. In terms of who is doing the work and who is involved, that can also be critical. At the early stage, these may be investigators. It could also be a pharma company at the beginning. You might want to found a startup company, for example, create an SME company that does some of the early regulatory engagement, for example. But to do some of these studies, especially clinical studies or even tox studies, you're likely to need someone to pay for that, such as venture capital, maybe a big grant if you're lucky, something like that. But usually at a later stage, a bigger pharma company is going to come in, license your product, and keep the development going, especially for phase 3. So again, this is for small molecules, but we'll have a look and see how this overall scheme works for ATMPs, okay? So the first example today is Strimvelis. So we're going to go a little bit into the background and then go over what the product is and how it was developed and some of the key aspects that came up during the development, okay? So all of this information, by the way, is in the public domain. This is not any Asphalian client information, of course, that we're presenting here. And some of it, of course, is also based on our experience and interpretation of what we've seen in the public domain. So just to keep that in mind. Okay, so Strimvelis is designed for the treatment of ADA-SCID. So SCID may be familiar to you from the mouse model, which is immune deficient mouse model. So SCID itself is a kind of group of inherited disorders based around that concept of immune dysfunction. So the two main types are ADA-SCID, which is caused by a mutation in the adenosine deaminase or ADA gene. This is responsible for about 10 to 15% of cases of SCID, and this results in impaired development of functional T, B, and NK cells. Okay, another key type is called X-linked SCID, which is caused by a mutation in IL-2 receptor gene, which is located on chromosome X. For this reason, only males usually are affected, and it causes a low number of or complete absence of circulating T and NK cells and loss of B cells. So you basically, in both situations, have a complete lack of functional immune system. So this is why the clinical features are basically lymphopenia, recurrent infections that may be fatal, and obviously this is associated with the classic boy in the bubble kind of scenario where infected children end up having to live in immune isolation because they essentially have no immune system. So even a small infection can be very serious or fatal. They also have other complications, non-immunological issues, developmental issues, and some behavioral and other issues associated with skeletal muscle and the liver. So this is a very rare disease. It has an incidence from about 0.2 to 0.68 per 100,000 live births. Usually in this disease, the incidence is often reported more than the prevalence, so the prevalence does exist. And one of the reasons for that is because usually you don't live very long, so you actually don't have a very different prevalence from the incidence in many cases, okay? So as we mentioned just now, that most of these patients are dying within 1 or 2 years of birth, unless you can somehow restore the immune function. Okay, so the causative gene for this disease, ADA, was identified only in 1972. So basically, it works like this. The ADA gene is on chromosome 20, and it's expressed in all cells. It's a kind of housekeeping gene, which we'll refer to a little bit later. And it's basically involved in enzymatic reactions and getting rid of accumulation of small molecules that are involved in energy metabolism, for example, and just general cellular metabolism. So the absence of this enzyme basically causes an accumulation of toxic intermediates in these cells, in this case especially immunological cells, and basically causes these cells to die, or they don't develop or don't proliferate, and so on. In other tissues, it's also the same mechanism that causes other tissue loss. But for our intents and purposes today, this is about the immune cells which lose function because they haven't got the ADA gene, and they basically just get toxic. So the current treatments for ADA-SCID are bone marrow transplant. So at the current estimates, you get an overall 6.5-year median survival rate when you have a very good HLA match. The reality is that most people won't have a good HLA match, and the outcomes will be much less positive, so less years, or with lots of other issues such as immunosuppression and so on for the host cells, or graft versus host disease. So not really a good treatment. Enzyme replacement therapy, or ERT, has also been around for ADA-SCID since the 90s, at least in the USA, because this product Adagen wasn't really approved in the EU, though it was available on compassionate use basis. And we'll refer to compassionate use a bit later in the presentation as well. There is a new product called Recovi, which is this one here, that was approved in 2018, but that doesn't have a real bearing on the story today. But one of the classic problems with ERT, or some of the problems, are immunogenicity, because you're basically injecting a foreign protein on a weekly or bi-weekly basis, so it causes a lot of immune reactions. It's also very expensive. It can be about 200,000 euros a year, I believe, and it requires chronic administration, so you always have to keep administering this product. Okay, and obviously gene therapy is now one of the new current treatments since Strimvelis was approved, okay? So before we go into some of the regulatory details, it's worth just revising a little bit some of the rationale for developing a product for this disease. Obviously, there's a huge unmet need here, which is one thing, and it's a very serious condition caused by a single mutation, so that makes it a little bit more amenable to being treated by gene therapy. So we mentioned before the ADA gene is a housekeeping gene, which is a good thing for this because it means you don't need to have fine regulations. So by that, we don't mean very smooth, let's say, promoter control. You can use almost any promoter that will express either in the cell that you want or maybe even ubiquitously, and you'll have very limited risk of having off-target toxicity due to expression of the transgene. So that is a very positive thing here. Another one is the ADA-CDNA is very small, so it's only 1.5 kb. In the previous presentation, we mentioned some of the problems with AAV vectors that can be very small, for example, for big genes. So this is a positive here as well. Another is that you don't need very high levels of the enzyme to have a normal function, or at least to have some beneficial effect, which is good because you might get an effect from less dose or less efficient grafting, for example. The other thing is the corrected cells or normal cells in the case of a ADA patient who has been given a bone marrow transplant, for example, these cells have a selective survival advantage over the ADA deficient cells. We already mentioned that they accumulate these metabolites which have a toxic effect and prevent proliferation, for example. So if you correct that, the cells that are corrected will go on to prosper and thrive and eventually overcome and replace the deficient cells. So a key concern here as well, which comes up in quite a lot of gene therapy applications, is do you or do you not stop the standard of care at the time of administering gene therapy? And in this case, it was found for ADA-SCID that it was necessary to discontinue the enzyme replacement therapy prior to giving gene therapy, even though that is in a way partially unethical. And the reason for that is basically because you lose or diminish that selective survival advantage. So you actually work, even though you're treating the patient, you're actually working against the long-term treatment from the gene therapy in that case. Okay, so the other thing, the target tissue here, which are hematopoietic cells, so the immune precursors from the blood, that is, so these are easy to get and culture and to transduce. It's not like you have to transduce the whole muscle, for example. So in this case, they're also autologous cells, which means you eliminate graft versus host disease, which we saw was a big problem with the transplantation approach. And also here, ideally, this is a single injection. You make one correction to the cells and they last throughout the lifetime of the patient, which is a considerable advantage over enzyme replacement therapy with chronic administration. Okay, so here's a little bit of a timeline. You saw a couple of these in the previous presentation, a bit of historical context, and also here I've mapped out some of the key regulatory activities that were done for the marketing authorization of Strimvelis, okay? So starting back at 1990, these two time points I presented in the previous presentation as well. So the first clinical gene therapy trials were actually conducted in this indication for ADA-SCID and published in 1990. So this is one of the most historical products in the gene therapy field, which is one of the reasons also why I chose to put it here. So you can see at the same time as it came out, the active control, let's say, the enzyme replacement therapy also came on the market in the US, okay? So looking forward then, there's obviously a big gap here. There was a lot of clinical trials and things being done through here, but there was some issues that came up, as I mentioned, in 2003 with the insertional mutagenesis in the SCID-X1 trial. So as you saw in the intro, this is obviously a very related indication, a very similar approximation, and I will show that in the next slide before going on to describe the procedures. But looking a little bit over here at the global view, you can see the marketing authorization was presented, 2015 and approved 2016. So all of the key regulatory activities are sort of clustered around the last stretch. So in yellow, I've put different protocol assistance procedures that were done, because obviously this is an orphan drug. The orphan drug was designated in 2005, right at the beginning of the whole process. You'll notice the FDA orphan designation was designated several years after that, usually because orphan designation can be done at any time in development. There was probably no rush for them at that time. Okay, so the protocol assistance, there was 5 done. 2 of them were done almost back to back at the end. This was possibly also to discuss the contents of the marketing authorization dossier, maybe some CMC issues, and also related to the ODD was a significant benefit discussion to make sure all that was okay prior to submitting the dossier. I also put here in red some aspects related to the Paediatric investigation plan, which I'll go into in a bit more detail. So here they presented a PIP in 2012, they had a decision in 2013, and they did a modification of that PIP right before they submitted the marketing authorization dossier. So we'll go through a little bit of that and look at some of the other aspects related to the evaluation of the marketing authorization dossier and so on. But first, I just wanted to go over quickly the insertional mutagenesis issue. So this was first reported in a clinical trial in 2003. So this paper here, which is now a seminal paper in the field because of the consequence that it had. So basically, the previous story to that was that 9 out of 10 patients with X1 SCID had been treated with a retrovirus that was replacing the gamma chain of the IL-2 gene. And they had really good response. All the 9 out of 10 patients were basically cured, let's say, of the disease. But 30 and 34 months after they reported that these patients had been cured, 2 of these patients, P4 and P5 at the time, was shown to have insertion of the retroviral vector into the LMO2 gene, which is a kind of oncogene or proto-oncogene. And it was believed to be that some of the viral sequences, the ITR, for example, was activating the oncogene and therefore causing clonal expansion of cells in which the retrovirus had inserted into the oncogene site. Subsequently, in this 2018 paper, it was reported that 2 other patients from the same trial, P7 and P10, also had insertional mutagenesis. P10 had 2 types, one in the BMI1 gene, another one in LMR2, and another one CCND, which is a cyclin gene associated with cell cycle. So that's obviously not good, but what they found was that they could treat these patients with chemotherapy and basically destroy these clones without destroying some of the other cured cells, so that the patient would eventually revert back to being cured, but then the clones would appear again. So it was a repeated cycle of having to treat the clonal expansion of the insertional mutagenesis cells and trying to get the other cells to survive, okay? So that has some bearing on Strimvelis, okay? So here's a little bit of an overview of the product, okay? So Strimvelis consists of autologous CD4 cells, and these have been transduced by the retroviral vector that introduces the ADA gene into these cells, okay? So the indication that was given to the product was the treatment of ADA-SCID, of course, which is specific to the mutation type. So it doesn't cure X1 SCID, you'll notice. So this is also an important concept of gene therapy, and also for similarity assessments for orphan products, especially at the time of marketing authorization. There is a new FDA guidance, especially on that, for example. In this case, obviously, it's not the first line treatment. If there's no HLA matched patients, then you will do the gene therapy approach. Okay. The current marketing holder is Orchard, and I'll go a little bit through the change of sponsor of this product during the life cycle. So the formulation here is a dispersion, which is not one you commonly see, but it's used for cell therapy type products, and this is for intravenous use. So this is obviously ex vivo gene therapy. So here, I just wanted to go through something that happens quite a lot. I mentioned in that one slide overview of small molecule development that you might have investigators starting off in SME or so on, and then coming in to license the product. And this is just to highlight this for this product. So it was started in Italy at the Fondazione Telethon in Milano, and it was initially the initial orphan drug designation was made in the name of that sponsor in 2005. The product was subsequently licensed to Glaxo in 2010, and indeed this was one of the reasons for some of the scientific protocol assistance procedures that were done around that time and around here. So that obviously if the change in sponsor, the different procedural procedures have to be transferred as well. So transferred originally to Glaxo in the UK, maybe because of Brexit that was subsequently again changed to Ireland. And then the product was approved as an orphan product in May 2016 by Glaxo, but the sponsorship was then subsequently transferred again to Orchard Therapeutics in Netherlands. So Glaxo is still a big sponsor of Orchard. So basically it's still their product, but it's under the Orchard brand. So that would be a pretty typical change of sponsor and change of a financial entity behind an ATMP development, for example. So looking at the actual marketing authorization application itself, there's actually very little to report of interest actually, but basically a couple of things. Centralized procedure, the eligibility was confirmed quite a long time before the MAA. So even though ATMPs have to go through the mandatory scope of the centralized procedure, the eligibility application still has to be made as a formality. So that was obviously done before just to assure that that's okay. The procedure was basically as standard, a little bit of validation. The valuation started, list of questions to the sponsor. These were then responded to. There was a list of outstanding issues and they were addressed quite easily. The CAT makes an opinion, and not long after that, the CHMP makes an opinion, which is endorsed by the European Commission. So it's worth mentioning here that there was no prime application for this product. No conditional approval was used, and there was no other accelerated assessment. So Lydia will give a talk about these themes in a couple of weeks' time. But the real reason for this is, one, prime didn't exist at the time. That only started in 2006. Conditional approval was not really needed here because there was no surrogate biomarker involved, and accelerated assessment does not appear to have been used because the review was relatively straightforward. This may also be facilitated by the relatively large number of protocol assistance procedures, which was about 5. There was also a very rapid pricing and reimbursement procedure that was done in Italy, apparently, at least according to this paper too. So that's also interesting to consider to get on the market. I wanted to talk a little bit about the pediatric investigation plan here. It's a little bit unusual for a couple of reasons, but I think it works as a good model just to describe some issues that can come up with pediatric investigation plans. So if you remember, ADA-SCID is usually diagnosed essentially at birth, so it's basically a purely pediatric indication. But what you'll see here, I will go back to this slide, you can see it easily. So the clinical trials obviously started here, but the real ones were starting a bit later here, as we'll show you in a later slide. But the PIP was actually done considerably later. So looking at these dates here, you can see that. So the initial PIP was a straightforward procedure, start the procedure in 2012. It finished more or less a year later, which is a standard PIP review time in March, with some request for modification and so on during the middle. The sponsor subsequently modified the PIP in 2014. Again, a relatively simple procedure. It's not always clear what measures and timelines were modified because they don't give that information publicly. However, one thing that you can see is on the initial PIP decision compared to the modification decision, there was a change in the date. And I think this is very important and attention grabbing here, because they basically started the PIP in 2012, originally saying they were finishing it in 2011, and then they modified the PIP here to say that they actually finished the PIP in July 2011 and not June. So why that is highly likely to be due to compliance. So before I say what that is, just go back a step. So when you do do a pediatric investigation plan procedure, you will have a compliance check at the time of submission of your marketing authorization to see that you are compliant with the pediatric legislation, which means you will either have a waiver, all the deferrals, etc., in your final opinion, and that anything that you had as an obligation to do before marketing is done and is cross-checked for compliance. So basically, and we've seen a couple of cases like this in Australian, that some companies get to the marketing authorization and then they realize, oh my, we've got some slight differences between what we said we'd do and what we did, and that can be something as trivial, it seems, as a date. So it seems here that what they've done is change the dates and maybe some other measures in their PIP just so they can pass the compliance at the time of marketing. So again, if you just go back here, you will see that the modification was done right before the marketing authorization, essentially. So this appears to be a purely compliance-based change. So the other thing that's also important here is they've tried to get a waiver at the beginning for the very young patients. This was rejected by the PDCO, and they were basically got enforced to do include patients from birth until less than 18 years of age. And they had a single study proposed in their pediatric investigation plan. As mentioned, this was finished already at the time of presenting the initial PIP. One of the reasons for this is probably because the pediatric legislation came out after the pivotal study had started. So the PIP was necessary for compliance at the time of marketing, so retrospectively they had to negotiate a PIP. This is obviously highly unstandard and not recommended at all, because if they don't like what you're doing, then you may have wasted your whole pivotal or key study. As well, this is obviously a historical development, so if you were to try that today, I imagine you wouldn't get very far with the pediatric committee. Okay. So here's a little bit of an overview of the manufacturing and the treatment for the patients before we go into some specific regulatory stuff. So here there is really two active substances, which is according to the current guidelines. So you have a retrovirus, which includes the ADA gene. So this is made by standard procedures and just frozen away. Then when a patient comes, you can harvest the bone marrow and use that as a starting material to manufacture the CD34 active substance. So you get the bone marrow, transfer it in from one container to another, start QC testing, send it to a manufacturing facility, start to isolate the cells that you want under GMP, transduce the cells with the virus. The virus is not specific to a patient, but obviously the cells are autologous, so they're specific to a patient. You then concentrate the cells, wash them, resuspend them, and then put them in a transfer bag, and then it becomes the drug product, and you send it to the patient. And this is how it's kind of administered. It's not clear here if this is steps or days. Usually the transduction process may take a little bit longer than a couple of days, but it may be that's just days. Obviously, they're keeping commercially sensitive information out of the EPA, but they also use busulfan as a preconditioning agent, basically to wipe out the endogenous cells of the patient before giving the autologous corrected cells. Again, this just confers more survival advantage to the patient. Okay. So looking at some of the issues that came up with the product during the evaluation. Okay, so they have two active substances. One is the replication deficient gamma retrovirus. So this virus actually has a neo-R selection marker. So this is not really very common to use. I don't know if they would still really want you to use that. It may be a historical thing, but the marker is basically just to allow selection in patients or out of patients if necessary, more out of patients for assays and things, I imagine. Okay, so some of the issues that came up were about comparability. So obviously there was some change in the manufacturing site or process, especially to get ready for commercial. So, or at least to go from the investigator phase into a more commercial ready product. So there was a lot of comparability stuff that was done there. One of the novel studies that was asked of them was to actually demonstrate the integrity and the stability of the provirus. So that basically means once the virus has infected the target cell, does the inserted sequence come out? Does it rearrange? Does it get mutated? How stable is that through different proliferations of the cell? So this kind of relates maybe even to starting material or cell bank stability issues that come up with gene therapy or advanced therapies as well. Okay, so for the second active substance, this is now the transduced cells, so the corrected cells. Obviously, compliance with those cell and tissue procurement and testing directives are critical. That should be done as part of due diligence of setting up a manufacturing facility and should be documented as well. Here, because of the type of product and what was involved, they actually had 3 potency assays. So they had a functional assay, which was looked for the activity of CD34 cells, so a pure biological assay, which is the pure definition of potency according to the guidelines. Then they had an assay for vector copy number, so obviously just to control that each infection is giving roughly the same number of integrations per cell, and then assay for transduction efficiency to make sure there's nothing else going on there as well. So again, they had to do comparability here. They had to do a side-by-side analysis of the commercial process for the cells with a commercial virus compared to the clinical process and the clinical virus. So here again, the implication is they appear to have changed the manufacturing process after phase 3 and prior to submitting the marketing authorization application. Just to jump back again to this slide here, it's highly likely that all these protocol assistants at the end were basically to talk about this issue, because that's obviously a massive aspect. The other comment that's also quite interesting here for this type of product is that there seems to be a wide range of specifications. So the values for the acceptance criteria are quite divergent to what you would normally expect, which is something you might accept in this case, given the low number of patients and the type of results they were getting as well. Okay. So for the non-clinical, just to go through some of the key concepts as well. Pharmacodynamics, like CAR T cells, basically the bulk of all the studies in the non-clinical section are done with pharmacodynamic studies in vitro, also in vivo with different models. Obviously, you have issues here trying to get donor cells. You can't really get ADA-SCID cells very easily in this case, though obviously they did some studies there, but justifying why healthy donor cells are fine or other SCID cells are fine, or even animal SCID cells are fine, for example. So that's a really important aspect here. This is also where the dosing information comes from in these kind of studies, also for CAR T cells. So for pharmacokinetics, there was a couple of interesting things that they were required to do here too. Drug-drug interaction studies. So we mentioned here that busulfan preconditioning was used to basically favor the selection advantage of the corrected cells. So they were asked to do an interaction to see that this was also not affecting the transduced cells that were introduced. Obviously had to do biodistribution. They did that with transduced and non-transduced cells because there is a risk that the transduction will also change the biodistribution of the cells. They did see some vector distribution, so in the gonads. So this is relatively common for gene therapy products actually to see some viral signal in gonad tissue. It doesn't mean it's transduced germ cells, for example, but there is a risk of germline transmission. So they had to make a justification of that in this development as well. Here they also justified not doing non-clinical shedding studies because basically, and also clinical, because the vector is used ex vivo, there is expected to be almost no vector in vivo. But one of the key arguments for justifying that is that you have no recombinant virus detected in the manufacturing process. Okay, so with toxicity, they did do some toxicity, a single dose toxicity with healthy human donor material. They couldn't do a lot of tumor immunogenicity studies because they couldn't get long-term engraftment of the healthy human donor material in mice. So this is obviously relating to that insertional mutagenesis issues. So they clearly have looked at that, but there's not really a good way to model that in vivo. So that's still an ongoing concern for this product, and I'll mention a little bit more of that later. So here's just a quick overview of their clinical development plan as well. So basically, here's the studies they've done. This one in italics is not really counted as part of the studies. This was a named patient program study that probably occurred between the time of submitting the MAA and getting the approval. But basically, they started off with these two early pilot studies. This was a one-patient study done in Israel, and these were two done with the Milan sponsor, and this is two patients done in Milan before the pivotal study was started. Okay, with only 12 patients. During that time, there was 3 patients who couldn't be treated in this study that were put into a compassionate use program. So these also formed part of the final evaluation. So there's 18 patients basically in the final safety database and efficacy database. 17 of these went into a long-term follow-up study. So this is also something commonly seen with gene therapy and advanced therapy clinical trials. So just having a look at that in another way, here's the same timeline as you have before, where the different procedures were done, and this is where the trials were done and started. So basically you can see the first studies were started very early, but had a long-term follow-up already of the patients. In parallel, a pivotal trial was done. This design was also agreed with the protocol assistance, or some changes were agreed in the protocol assistance. And the PIP was started essentially after already the pivotal was done, as we already mentioned, so to retrospectively include it. The long-term follow-up study comes after the pivotal study, so all of these patients need to go into that study. So this is also logistically an important issue, because basically it means this study design needs to be active even when this study is beginning. So probably now you would expect almost them to be submitted at the same time. Why? Because maybe you need to have that open for the first patient to come off here, because that duration is maybe exaggeratedly long to some degree because of the follow-up that's been done, but also because of the rarity of the patient. But the first patient, if it was treated at the beginning, would already need the long-term follow-up study here. And obviously the compassionate use is done in parallel. I've also mentioned here the registry study, which was a requirement of the marketing authorization. Again, this is highly typical of an advanced therapy medicinal product. This study is due to go on until at least 2030 or 2035, something like that. So just a quick overview of the clinical development plan. No formal dose response studies here. There was some data, of course, presented in the dossier. So this is similar for CAR-T kind of cell thing, how much transduction you're getting in the manufacturing process, how many cells you're going to infuse, and so on. So an upper and a lower limit usually needs to be set for cell therapies, but that was done usually based on animal and non-clinical studies. Another thing done here was that patients had their bone marrow harvest, and some of it was put aside as a rescue therapy. So if everything goes wrong, they might be able to chemoablate the cells and restore at least some of the bone marrow to the patients in case there were safety issues. Another thing here being an ultra-rare disease, and also because the manufacturing site is also essentially linked to the clinical site, there was only one site involved in the study. So this is kind of good because it ensures that everything runs smoothly and you get consistency of the results, but then there is always the doubt about whether this data is specific to that center and could be reproduced in another center. In this case, because the manufacturing is in that center, or at least close by, then the 6-hour stability of the final drug product means it's almost impossible to send it to another place, which means all the patients will have to go there, or that they will have to validate manufacturing in different sites for use in trials or in other countries, okay? Another key thing here was this, because some of those early studies were done under academic guidance, let's say, they were not always done to the best of GCP, and there was some GCP failings there. So that could also be an issue, especially for historical studies, but most ATMP studies going on these days should hopefully be done according to GCP with all the right documentation and follow-up and so on. One of the key things was obviously about the insertional mutagenesis. So they were asked to do a retroviral insertion site analysis and to develop a specific assay to follow this up in these patients. To date, and I haven't seen anything to the contrary, but there was in the marketing authorization, there was a medium of 7 years follow-up from those patients, up to 13 years for one, which is quite a lot longer than the 3 years or 30, 36 months that was seen for the X1 SCID patients. So that would suggest, at least at this stage, that there is no insertional mutagenesis, or that it's not at least a really dominant form of insertional mutagenesis in these patients. They were also asked to do the non-inventional post-authorization study, i.e. the registry study. So they need to look for 15 patients for a minimum of 15 years, including immunogenicity assessment. So this is pretty standard as well. The guidelines do suggest 15 years at least should be followed. I mean, that can be negotiated in some cases, but it's usually about that. Okay. So in a little bit less detail, we're going to look quickly at Luxturna and see some of the things that they've done. So Luxturna is a gene therapy product for the treatment of inherited retinal dystrophies. So this is a very large group of inherited eye disorders that cause irreversible loss of blindness. So there is a very wide spectrum of presentations, and this it's a genetically heterogeneous set of conditions. So if you look at this figure on the right, this basically shows you some of the key IRDs, so retinitis pigmentosa, Leber congenital amaurosis, for example, macular degeneration. So these little numbers here give you the number of genes that are causing this condition, and where they overlap, you can see that the same gene causes different conditions here. So that's obviously a big issue, because you obviously have lots of phenotypic variation as well as genotypic variation. But basically, for this product, Luxturna, it has a RPE65 gene, so it's used to treat RPE65 mutations, which can cause LCA or RP mostly, but also other forms here. So this emphasizes you need not only a phenotypic, but a genotypic diagnosis here. So as mentioned, this is a gene therapy vector, AAV2 with RP65. It's indicated only in patients with inherited retinal dystrophies, which have a confirmed RPE65 mutation, and they also need sufficient viable retinal cells for this to work, because this is the target cell. So if you've also lost your target cells as part of the disease, you're not amenable to treatment, basically. This product is presented as a solution for injection. I highlight solution because actually it's probably a suspension, but for some reason in the EU it's called a solution. It's actually provided as a concentrate and is diluted down for intraocular use, so direct injection into the eye. The sponsor of this is currently Novartis, but it was developed by Spark, who came out of CHOP in Philadelphia. I'm not going to go into the provenance of this product this time. You've seen an example of that. So here's again our overall timeline, much simpler in principle than the ADA-SCID, probably because it's a much simpler approach overall. So a bit of context. So the first gene therapy in mouse models for this condition was done in 1996, big canine model in 2001. There was a huge flurry of studies and competing groups doing that in the 2000s, more or less. But the real development of this kind of started around then, I guess. So it's a much shorter development than for the ADA-SCID. In terms of the procedures, there's not really too much to say here, except about the Orphan designation, which I will go into detail. The PIP was done as well. As I mentioned, it's a mandatory requirement for marketing, so I won't go into that. They did only 2 protocol assistance procedures for this, very typical as well. But I'm just going to talk a little bit about the marketing and then the ODD and a few other key aspects. So in this case, the marketing authorization went exactly as for ADA-SCID, with one exception, which I've just highlighted here. So they did have some outstanding questions that could not be answered at the time of the list of outstanding issues that involved an extra oral hearing in front of the ad hoc expert group. So this is also something extraordinary that can happen in these kind of procedures, which, you know, to justify the efficacy, the indication, the use maybe, and so on. You can see the details in the EPA. Again, it's interesting to note that none of these procedures were used, but actually, again, this is a timing issue. This product was already submitted around the time that PRIME started. Again, no conditional approval was needed, and accelerated assessment of any kind was not done, as far as I'm aware. Okay, so from a quality point of view, again, there were some comparability assessments that were done because of the change in the manufacturer between the phase 3 and commercialization, exactly the same as ADA-SCID. This is obviously a big issue. Here they used a couple of PPQ batches to do the process validation and do the comparability. There was some concerns about that, but I've just put this to highlight how critical this is and how common it is to need to do comparability for ATMPs, basically. There was apparently some deficiencies in the CMC aspect. This is related apparently to the timing or the proximity of the MAA to the pivotal studies. So it doesn't look like it looks like quite a bit of time here, but it's actually not very much time, and we'll see the clinical plan as well. But basically, if everything is jumbled in together, it can be quite hard to get everything up to commercial standard in time, and that's really one of the key messages of this presentation today, that ATMPs have shorter development plans, but that can cause problems at the back end, so to speak, on CMC and other aspects. They also had some issues regarding the US manufacturing site. So this is manufactured in the US, but imported into the EU. So it's worth highlighting this as well, that according to the GMP Annex 16, which is about QP release, there should be retesting of this product in the EU before it goes onto the market. This can be waived or reduced, and there's some flexibility allowed by a mutual recognition agreement, but the MRAs that are in place between the US and the EU do not cover ATMPs, so you're left out a little bit on the cold there. But there is some aspects in the GMP annex for ATMPs that allows for flexibility there, but that issue is important and should be looked at if anyone's manufacturing in a third country and importing into the EU. I'm skipping non-clinical here because there wasn't really much of interest, let's say, in this dossier. I wanted to highlight the clinical plan again because it's, again, quite interesting here. Again, there was only one pivotal study. In this case, it was a phase 3 study, not a phase 1-2, as in the ADA-SCID. And here they showed some durable improvements in visual performance for at least 2 years. So there was 2 supportive studies, the 101 study, which originally just treated one eye of these patients with LCA, so RPE65 mutant, of course, and this was just a safety tolerability study in 12 patients. Subsequently, the same patients were then treated in this study, but the other eye was done, except in one patient. So this was obviously because of safety concerns. It might have been the first time, maybe, excuse me, if that goes wrong, you don't want to knock out both the eyes at the same time, but then it was shown to be safe, so they then did both eyes in the pivotal study. They also had a natural history study here, which is another important concept for ATMP development, that these kind of studies are critical for supporting sometimes the lack of controls in the pivotal studies, okay? So this is how the study, or the clinical study plan, was done for this product, okay? So the Orphan designation came in quite late. They had 2 of them, which I'm going to talk about in a slide in a minute. They had the first study, which was quite long, probably for recruitment and getting follow-up and so on. So these patients went into that other study, as I mentioned. Then the pivotal study came along, which was much shorter, presumably because there was a lot of interest in these products coming in. The follow-up was much more controlled and so on. It's also worth noting, I've indicated here a little bit how the design works. Here, these patients have a run-in of 1 year, so the patients are followed for 1 year before they're treated as a kind of control within the same patient, within the same study. This was also a controlled study, which means that those who were given placebo were actually not treated for 1 year as well, and then treated afterwards. So that way you can get some kind of less bias, let's say, in the clinical trial design. And after that, the extension is done as a separate study. That's always done like that, to stop this study, get the CSR, get your marketing done, and then do the long-term follow-up, as long as you have a minimum long-term follow-up. Okay, so here, just before we finish off, I just wanted to talk about the orphan drug designation for this product, because like the PIP for ADA-SCID, this is a really interesting example of complications that can come up and how the indications and so on are defined. So here they actually requested two orphan designations. So the first one was for the indication of Leber's congenital amaurosis, obviously caused by RPE65. And as we mentioned at the beginning, the same gene may cause a different condition. So subsequently, they also requested orphan designation for retinitis pigmentosa caused by RPE65 mutation. Around the same time, they were doing the PIP, which had a different indication, which was genetic congenital retinal disorders. So this is probably because of the way they assign the indications in the PIP as well as the orphan condition. This indication here is more like the final indication that they ended up with, and also relates to the change in the orphan designation as well. So when they requested the marketing authorization, they requested the indication treatment of patients with LCA, Leber's congenital amaurosis, or retinitis, which are caused by confirmed RPE65 mutations. But when the review was being done, the comp then insisted that this is not the case. They have two orphan conditions, and they went on to say that the condition should be inherited retinal dystrophies caused by RPE65 mutation. So that means then the clinical development plan, as you remember, was done in LCA caused by RPE65. So this is an indication that falls within the conditions. So that means it's amenable to the benefits of the orphan designation. Okay. And this is also important, again, for the similarity exercise for orphan products and ATMPs, as especially defined in the FDA guidance, right? So here's a bit of an overall summary then of those two products. So usually the ATMP path to market is abbreviated compared to small molecules and other biotech products. So this creates a lot of CMC pressure. As we've seen, there was some problems with some of these products. This may be method validation requirements are early, process validation are early, for example. You may also have potential GCP issues, logistical issues related to that acceleration as well. And in all of these cases, comparability studies were done. And that should be really something considered from the outset for an ATMP development, that it's likely that some kind of comparability or change in manufacturing will be required, okay? So many ATMPs are developed for rare pediatric diseases as well. For that reason, the timing and maintenance of the PIP should be done very carefully and with consideration, and to not delay marketing, it should be reassessed beforehand so that there's no compliance issues. Just as on an aside, the Luxturna product also received one of the priority pediatric review vouchers from the FDA for their work in this indication. So that's also a nice reward to have. The orphan designation you've seen can be done at any time, and this is also something that's reassessed at the time of marketing. It's not a static thing that you get and it gives you something forever. You also need to maintain that and look at that carefully at the time of the marketing authorization. And here, regarding accelerated assessments, they were not used in these two models, but they are available and should be used where possible. Okay. In terms of clinical aspects, you might not be able to do good dose response data. Maybe you have one dose level, something like that. So that's something that should definitely be discussed with the regulatory agencies again at the beginning of the clinical development plan, because you don't want to get down the track and then just find out that you're not using the right dose. One pivotal clinical study may be a feasible approach, especially in some of these orphan conditions, but there's needs to be very strong data, especially if you have small numbers of patients in there. Long-term follow-up studies are always required, so you should expect to do something there with either as a clinical protocol, as a PAS registry study, for example. Also, because of the type of product and indication, you also get these other less common types of situations where you have a compassionate use program, named patient program, and also natural history studies coming into play. So really clinical development for ATMPs can be very polyvalent compared to others. I already mentioned some of the other upcoming webinars. So Joelle's going to talk about the procedures I mentioned today. John's going to give you a summary of vaccines for COVID, and Lydia will talk to you about some of these expedited pathways that I've just mentioned here for ATMPs. So there may be some questions as well. If you do, put them in the little box on the side. If not, thanks for listening, and we'll see you at one of the other webinars. No questions today? All right then. Thanks very much for your time, and yeah, look out on YouTube and make sure you register for the other videos. So be safe and see you soon.